Okay, welcome to the Exam 3 review. We're going to go until we're done. I will cover the most important parts that you need for this exam. The final exam, of course, is coming up soon as well. This is going to be recorded and posted in parts like the other exam reviews, so I'll do that as quick as possible and put it on YouTube. What else to mention? Oh yeah, if you haven't heard yet, this is going to be my last semester, so if anyone is thinking, yeah. Anyone wanting to take up the mantle of Biochem SI or whatever, feel free to. We need people to instruct this. If you're interested in that, come talk to me. Some of you have already talked to the CTL about that. If you are also interested, let me know. Other announcements stuff. Oh yeah, I won't be here on the day of the final exam, but don't worry, I'll still be able to give you guys the exam reviews and everything for the final exam. Don't go in there thinking that you'll have me to ask like questions on a bunch of stuff on the day of the final exam. All right, but don't worry, I'll try to prepare you guys as best as I can. Other announcement, if you haven't heard this, 29B, meaning eukaryotic transcription, will not be on exam 3. You're welcome. The exam 3 will only be, oh my god, that door. Will only be 10A, 10B, 11, 28, and 29. All right, so 5 chapters worth, yes.
They're studying eukaryotic transcription in 29.
At the end of 29, yeah, so
We don't have to know that.
29A, I should say, yeah, so don't worry about eukaryotic transcription. Let's see. All right, do not worry about this. Other than that, I guess I will get started, and we'll start, of course, with 10A. Let's see. From the beginning. All right. So, what was the important part of 10A? 10A was to introduce you guys the concept of the central dogma and the components of the central dogma, which really is the nucleic acids. So, okay, I'm not doing that. Let me just scroll down.
You said,
So,
sorry, you said it was eukaryotic transcription?
yeah, eukaryotic transcription will not be on exam 3. Eukaryotic transcription is going to be on the final. So, central dogma of molecular biology. I'm not going to go over it in detail because it's pretty straightforward. You guys should have been studying it already. Basically, there's a couple different components here. You have DNA, RNA, and protein. And there's DNA, replicates itself. DNA is able to transcribe itself. You're able to transcribe DNA into RNA, and you can translate RNA into proteins. So, 10A's goal, the goal of 10A is to go over nitrogenous bases, nucleosides, and nucleotides. You do need to know the structures and what is a pyrimidine and purine for these nitrogenous bases. So they're listed here. If you want pictures of them, they're on here as well. I've gone over some shortcuts to help you guys memorize one picture from another. You can review your notes on that, but a shorthand for that is if you look at your pyrimidines here, cytosine, I personally just memorize it as having this amino group right there, and I kind of forget everything else. Uracil, instead of an amino group there's a carbonyl, and thymine looks exactly like uracil, except it has this methyl tail there. Your purines are those 2 membered ring pictures and structures here. You have adenine, which I remember as having that amino group there, and I forget everything else, and guanine, which has a carbonyl group there instead. So you do need to memorize these structures and be able to identify one from another. You may even be given nucleosides or nucleotides with these nitrogenous bases, and you have to correctly name them. So, don't worry about RNA bases. Those are really special bases that are rarely used, so don't bother memorizing that. Properties of nitrogenous bases, really the only one you may run into is this slide right here. This slide describes how you can identify RNA from DNA by measuring UV absorbance. And honestly, if you don't really understand it, this phrase here is all you need to know about it. This is an experiment we do in the lab to identify if a sample, how pure it is with RNA or DNA. We measure a ratio of 260 to 280, absorbance ratio at 260 to 280, and these numbers represent pure DNA and pure RNA respectively. Don't worry about the graph. Moving down, of course, after you talk about your nitrogenous bases, you have to talk about your nucleosides, and it's very simple. Nucleosides are just the nitrogenous bases with a sugar, and it will be ribose if we're just talking about nucleosides. So, make sure you know how to name them correctly. If you want all the names here they are. Cytidine, uridine, adenosine, guanosine, and not shown here, but thymidine would be another one. You do need to know the one that has bioactivity, which is listed on here as well, which is adenosine is the only nucleoside that has bioactivity. Remember, the other ones need to have phosphates or stuff attached to it in order for them to have bioactivity, but adenosine is the only one at the nucleoside level that has bioactivity. Now, these next few parts, before I have told you guys that you just need to know the concepts for the exam. Brand new this semester, you may need to be able to identify these compounds here, which is not that bad. Azacitidine, you will have to know what that does, and we know it is a drug to treat what disease? Acute myelogenous leukemia, which is going to be on like the next 2 slides, all right? If you're given a bunch of pictures of nucleosides, this is the one that is azacitidine, okay? You may have to come up with some fancy way to memorize it. I'm not going to bother. I'll leave that to you guys. You guys are good at memorizing. That's why most of you are pre-meds or are planning on going into those other pre-health fields. So I will leave that to you guys. Don't worry about the mechanisms of azacitidine or anything, just that it is there to treat this disease, acute myelogenous leukemia. Going down, going down, going down, going down, going down, going down. Where's the next one? Here we go, gene therapy. So, other nucleosides. There is a nucleoside that you can design, and here's some names of them. These you won't have to memorize their structures for. Just know that these nucleosides work as antiviral drugs. The way they work is they specifically target the virus version of an enzyme called thymidine kinase, and they're very specific. They end up ignoring the human version of thymidine kinase, so they're a very good drug compound. Oh, this one's interesting. ICAR, I can almost guarantee you this is going to show up on the exam in some fashion. You need to know what it does and what it looks like. Again, previous semesters that wasn't important. This semester it might be important. So make sure you know what ICAR looks like and that it is a mitochondrial production factor. It's a performance-enhancing drug, of course, so if you want to cheat at some athletic event, you can take this. Nope. Okay, yeah. Nucleotides. So after your nucleosides, you have your nucleotides, which are just a slight more complicated version of nucleosides. They are nucleosides plus phosphates attached to them. There are 2 categories of nucleotides that you can have, ribonucleotides and deoxyribonucleotides. And there's some, if you're given any one of these pictures, you have to correctly name it. So same with the nucleosides, same with the nitrogenous bases. If you're given any one of these, make sure you name it correctly. It's really easy to name a nucleotide. You just name the nucleoside part, plus tell me how many phosphates are on there. Monophosphate, diphosphate, or triphosphate. A nice example up here is adenosine monophosphate. It's adenosine with a single phosphate attached to it. All right. The functions of the nucleosides are very important. That's really just on this slide, so don't stress hunting down the functions of nucleotides. ATP is important for metabolism, GTP is for proteins, CTP lipids, and UTP is carbohydrates. Let me check something real quick. And this thing that I have in my hand here, I do not have. This does not exist. What I have right here, that is nothing. Okay. Yeah. All right. It is nothing. So, Pyrimidines and purines, I want to mention that you should be able to explain what a pyrimidine looks like or a purine looks like in multiple choice format. So single ring, double ring, that's really the most important component. Be able to tell me which one of these are going to be purines, which of these are going to be pyrimidines, and you'll be fine. Going back down to where we were, which is nucleotides. Functions. Oh yeah, cyclic nucleotides. Don't worry about memorizing the million different cyclic nucleotides. There's really only one that is talked about in detail. Cyclic AMP was talked about previously, but you don't need to know cyclic AMP for this exam. That's this thing. Cyclic GMP. That one is a smooth muscle relaxant, and it is a signaling molecule. There's plenty of details here. You really just need to know that it is a smooth muscle relaxant. All right, and the fact that cyclic nucleotides are signaling molecules. Another term for that is secondary messengers. All right. Although it isn't any one particular question, you guys should know that every nucleic acid is read in a 5' to 3' direction. So, if you're given any sequence on the exam, just in case I'll draw an example here. If either in the question or an answer choice is written like so, and there's no labeling, you must assume that the left side is 5' and the right side is 3'. Okay? That is a common mistake a lot of people make. They do their work for a particular question and they forget to flip their answer around to match the answer choices. Yes, question?
For nucleotides, can you have like the nucleotide?
The thymidine or thymidine-like monophosphate or something? Yeah, you can have. But I'm not aware of what that could possibly do, so don't worry about it specifically. All right, so again, make sure you know these directions. None of the multiple choice answer choices will have a 5' or 3' labeled for you, so you have to know immediately the left side is 5' and the right side is 3'. Okay. So, the classes of nucleic acids, again, this is general information. You should know this, DNA and RNA, and how the fact that there's a bunch of different types of RNA, and a lot of these you should know from other classes. What's unique for this class is siRNA and long non-coding RNAs, so make sure you know what they do. Oh yeah, let me make sure one thing here. Oh, so I get to pick that, okay. Anyways, yeah, so make sure you know the different RNAs. This is just a good illustration of your nucleic acids and how they connect to each other. The reason why we pick directions on a nucleic acid as 5' or 3'. The answer, or the reason, because it's not really an answer to anything, the reason why we label things 5' to 3' is that the phosphate at the very front end of a nucleotide or a nucleic acid, the phosphate is on the 5' carbon on your ribose or deoxyribose. And at the end, the very end of your nucleotide or nucleic acid, it's not really illustrated here, the very end on the 3' carbon is a hydroxyl group. You guys should know that when you build the nucleotides, you build it by adding new nucleotides on the 3' hydroxyl end of the preceding nucleotide. That's a very straightforward thing. All right. Yes?
Were we saying that's
Nothing.
okay? Cool.
Nothing.
Nice.
Nothing at all.
Sweet.
All right. All right, just, to poll, out of siRNA, lac operon, and prokaryotic transcription, raise your hand if your favorite topic amongst those three is siRNA. Raise your hand if it's prokaryotic transcription, and raise your hand if it's the lac operon. Okay, just a poll, all right? So, you can tell this is my last semester. Moving on from that, there is Chargaff's rule, which is the base pairing rules, right? Where the amount of A's in something should equal the amount of T's, and the amount of G's should equal the amount of C's. The way that this is applied, we've actually practiced this in SI, but I'll practice it one time up here because it's kind of the only calculation question you will get for exam 3. And that's something like, whoops, that is something like, I'm going to make up some numbers, I guess. In single-stranded DNA, you might have, and hopefully this adds up to 100, 50% A's, 20% T's, 10% G's. How much do I have left? 5, 6, 7, 8. Oh, that's okay. 20% C's. Okay, you will be given the information of how much of any of these bases you'll see in a particular single-stranded piece of DNA. And you can check the SI videos for how we solve this in detail, but shorthand explanation is, the question is going to be asking how much of each of these do you have in duplex DNA? All right, how much of each of these do you have in duplex DNA? And again, duplex DNA is a synonym for double-stranded DNA, so the way I solve it personally is I draw out the single-stranded DNA, I figure out what the other stranded DNA has, and then I do some math. So, single-stranded DNA, I'm going to draw, right, 50% A, 20% T, 10% G, 20% C, okay? The other strand of DNA then, you can conclude the following. If the first strand of DNA is 50% A, this is going to be 50% what? T is good. This is 20% A, because base pairing rules. This is going to be 10% C's, and this is going to be 20% G's. So if it's asking how much A's, what percentage is A's in duplex DNA, you would add up that, and divide it by 2, because if you add it up straight up, this all adds up to 200%, which isn't right. So you add up these two numbers, you divide it by 2, that's how much you have in duplex DNA. There might be other ways to solve it. This is my personal way of solving these types of questions. You'll very likely get completely different numbers than this, but it's pretty straightforward, as you can see.
So you add the 50% plus 20 divided by 2?
Yep, exactly. You add up whatever base they're asking for, you add them up and divide it by 2, and that's how much you have in duplex DNA. Let me just check some things. Alright. There will never be any history questions. I don't think I've ever seen a biology course that ever asks you any history questions, so skip. Maybe there is. I personally don't like that. Let's see. Oh yeah, number of hydrogen bonds. Which is stronger, GC or AT? GC, because it has how many hydrogen bonds? 3. A's and T's have 2. You may not get an exact question that asks that, but there will be several questions where knowing that will be important to solving whatever it's asking. Some examples in past semesters and what we've gone over at SI is if you're given a bunch of different sequences, how do you know which one will form a strong cruciform? You first have to identify the cruciform, and then you pick the one with more G's and C's in it. Another one is maybe which one is a strong promoter. Well, we know for promoters, and we'll get to it more later, that you have to identify which one looks more similar to a consensus sequence. And this extra fact here, the better promoter is 1, it has to look exactly like the consensus sequence, and 2, if there are 2 answer choices that look exactly like the consensus sequence, the one with more A's and T's is better. Why is that? Because what is the purpose of a promoter? It is the part at which DNA is going to open up initially. Does that make sense? A good promoter needs to look like the consensus sequence, and I'll reiterate this when we get to chapter 29. It needs to look like the consensus sequence, and if you have multiple answers that look like the consensus sequence, the one with more A's and T's in the surrounding area is the better promoter, because it's easier to open up that DNA. Yes?
Will the consensus sequence be provided?
Yes. Good question. Don't worry about memorizing consensus sequences. That will be provided in the question. Yes?
So for the negative 35 consensus sequence, doesn't that consist of mainly G's and C's?
Yes. So what I mean is the surrounding
Yeah.
DNA sequences around the consensus
Like the
sequence.
hair,
So
what's it called, hair?
no, that's a different component. So remember that the consensus sequence doesn't take up 100% the area of the promoter. Consensus sequences are only like a couple bases long.
Yeah.
So you look at the bases around the consensus sequences. The ones with more A's and T's are easier to break open, so you can open up DNA a lot
So
easier.
around the consensus.
Around the consensus
Must be
sequences,
A's and
more
T's.
A's and T's will win. Yes, will be the better one. Yeah.
Would there be like a question like for the -35 sequence, if there's more G's and C's, if they're both the same, then?
The first step should be consensus sequence first. Yeah. So in order to figure out the better promoter, first should be, does it look like the consensus sequence? That should always be the first thing. Ignore the A's and T's part as your first step. Your first step is, does it look like the consensus sequence? Only when multiple answer choices look like the consensus sequence, you break the tie by looking at how many A's and T's are nearby. Okay? Because that indicates how easy it is to open up DNA. Yes?
There were a few times in class when Dr. Gacy's called the Britnell box the top.
Yeah, that isn't just Dr. Hayes. A lot of biologists will do that. And part of that is some type of old time notation stuff. For this class, for the exam, the phrase Pribnow box is only associated with prokaryotes, and the phrase Tata box is only associated with eukaryotes. So for the exam, it will be that specific. Okay. So again, for those who didn't hear, Pribnow box is a consensus sequence in prokaryotes. Tata box is a consensus sequence in eukaryotes. All right. So let me make sure I'm not forgetting anything yet. Oops. Oh yeah, all right, so this I'll talk about in 10b some more, but there are differences between prokaryotic mRNA and eukaryotic mRNA, lots of differences. One is illustrated at the end of 10a, and that is that eukaryotic mRNA must undergo a process called splicing, and the reason why is that eukaryotic genes have these intervening sequences called introns that will not be in the final mRNA product. These introns don't code for anything. They need to be removed out of the mRNA, and these orange boxes here represent the important stuff, the exons, the protein coding sequences. Those need to be spliced together, and then you have your finished mRNA products. So, the terms used to describe this, I will jump to 10b, is for prokaryotes, we use the phrase polycistronic mRNA. That's somewhere on here, right? Yes. Is polycistronic RNA, polycistronic genes, polycistronic mRNA, it all means the same thing. The phrase polycistronic is associated with prokaryotes. It will start with the letter P is how I memorize it. What it actually means, what polycistronic genes mean, is that the genes, and you can see it here, are all right next to each other. They will all be transcribed simultaneously and will all be on the same mRNA strand. So that is only on prokaryotes. That is only what the phrase polycistronic gene means, or polycistronic mRNA, is that a single mRNA is going to have multiple genes on it, and that is only prokaryotes. Contrast that, of course, with eukaryotes, which have monocistronic mRNA, and the reason why it's called monocistronic mRNA is that a single mRNA only has the information to code for one protein. Polycistronic mRNA, one mRNA can code for many proteins. Monocistronic is one mRNA, can only code for one protein at a time. Other things associated with monocistronic mRNA we had just discussed. You need to splice out stuff called introns and splice together stuff called exons. In 29b, you learn we even modify it further than that. Monocistronic mRNA, you gotta add a poly A tail and a 5' G cap. That's probably not going to be asked about on this exam, but you know, it's good to know that there are other modifications with monocistronic mRNA. Let's see. Yes.
So you can never ask for like the question based on the stability of mRNA during this process. I mean, because that would be from the
29B or something.
It could be the poly A tail or the 5'.
Yeah, there won't be anything on poly A tail stuff for this exam, so don't worry about it. Let's see. Oh, okay. Ribosomes. There's a lot of numbers associated with ribosomes. General idea here is that ribosomes are made of rRNA. That's what rRNA stands for, is ribosomal RNA. And the fact that ribosomes, the stuff that makes proteins, is made of both RNA and protein. All right, so very simple stuff here. Don't worry about memorizing, 70S, 30S, 50S, whatever. Don't worry about memorizing those numbers. Just what is a ribosome, what is it made of, what does it do, and then you're fine.
Just that protein is made out of rRNA.
Ribosomes, that's listed here, are made of both RNA and protein. Okay.
Josh, did you go over, did you finish 10A?
I will have to check it at the end, but yeah, those are the most important things. I'm going to go through everything first, and I'll go back and fix a couple things. But yeah, 10A, that's the majority important stuff that you need. Other questions? Okay. Yeah, it might be easier than you think. All right, so tRNA, you know the general function of tRNA. You'll be relieved to know that most tRNA questions will be asked in chapter 30, not chapter whatever right now, so save this for later. You don't need it for exam 3. siRNA, you may get multiple choice questions, you may get extra credit questions maybe. And if it's not an extra credit question, you should definitely know it enough for multiple choice. Yes?
What are the extra credit question options?
The extra credit question options were siRNA, lactoperon, and prokaryotic transcription. Those were the extra credit options. Yeah. So, all right. So, siRNA. You should know what it's for. siRNA is for gene silencing. What we mean by this is that both naturally and artificially, we can inhibit the expression of genes at the translational level. What I mean by this is, transcription can occur normally, but we can shut down gene expression by preventing mRNA from being translated. One example of doing that is using this stuff called siRNA. So there's a couple diagrams you can look at. Okay. There's a couple diagrams you can look at, one of which is this one. This diagram tells you how siRNA is made. Make sure you know the different steps in there. You can also review SI videos and stuff. But yes, questions?
So for the extra credit question, how much do we have to know?
What's it?
For the extra credit question.
Any extra credit question, you have to explain it to the level that it shows you that you know what you're talking about, which is most often steps involved and the components involved. And as long as you have everything, even if you don't have everything on there, it's fine. Just give me enough that it shows me you know what you're talking about. Usually the way I describe it at SI is as much detail as I require. Yes.
So just to clarify, once the double-stranded RNA is cut and then separated, it binds the native mRNA, and when it binds, that forms a risk?
Yes, exactly. What is vibrating up here? Something is vibrating up here. I guess so. All right. Anyways, yeah. And you can find. All right. You can find that the second step in terms of siRNA is listed here is the single-stranded siRNA is going to combine with a bunch of proteins to form the RISC complex. Very important to know that there are two results that you get out of the RISC complex. That is, if the siRNA binds very strong, is high complementary to its mRNA, it'll cut the mRNA, and if it binds just okay, the RISC complex can still inhibit translation by binding to the mRNA and preventing the mRNA from being translated through the ribosome. There is potentially one other fact I gotta give you guys. 1 second. I believe. Nope. Okay. So
the native mRNA is cleaved if it's bound tightly?
Yep. If it's high complementary, which what I mean is that's what it means when it binds very tightly, it will cleave that. So those are two possibilities, two end results of siRNA. Other important information regarding siRNA is generally applications of RNA interference, of which you have a couple right here that you should learn. One or more of these might be an answer choice or so make sure you notice you know everything on here. Move it down. Don't worry about the actual crazy specific mechanisms like illustrated here. So I'm just going to skip all that. Let's see. Oh, yeah, I usually remember to talk about this in 10A. But this whole thing with cytosine and uracil. So, it turns out, for whatever reason, cytosine can spontaneously turn into uracil through a process called deamination, which can be problematic, right? Because uracil is not supposed to be found in DNA. So every now and then, the cytosines in DNA will sometimes turn into uracil. However, most of the time, we have repair enzymes that are constantly patrolling your DNA to look for whether something turned into uracil. And the way it figures this out is if it finds a compound that looks like thymine but doesn't have a methyl group, it knows that that's actually uracil. And it knows that what used to be there is cytosine. So it removes the uracil and puts in cytosine. So that methyl marker on thymidine is really important. That methyl marker tells repair enzymes that it's supposed to be there. The lack of that methyl marker at that spot means that it needs to be removed, and you put in cytosine. Yes?
So technically, is the thymine the marker or the methyl?
The methyl group on thymine is the marker.
So he wouldn't put like your liver.
It won't be that specific now. Let's see. All right, 1 second. This stuff, okay. So, you may be given a question that gives you a sequence, a single-stranded DNA sequence. A, T, G, C, A, T. And usually they will throw in a phosphate on either that end, or sometimes, to throw you off, they may even put a phosphate on the other end. That's still possible, not naturally speaking, but that is still possible if, like, this is a DNA fragment that was cleaved from some process, okay? So don't panic, don't panic if you see a phosphate on the 3' end and think that this must be some trick question. That can still happen if you cleaved this DNA fragment from something else using some other process, yes?
So we wouldn't have like a phosphate only on the 3' and not on the 5'?
It could be anywhere. You could have phosphates on both ends, you could have phosphate on one end or the
So
other.
it's just on the right end, and do you have to flip it, or can we still
Nope.
find the
If nothing's labeled, you still always assume 5' here and 3' over here. Okay, always assume that.
So, even though the phosphate is on the
Yep, and again,
still rules apply.
yep, the same rules apply even though there's a phosphate here, because usually a question like this will be given and they may ask you something like, the above nucleotide can be found in DNA. The answer is true because there's A's, T's, G's, and C's. There's no uracils. This thing might just have been cut off from something. Okay, don't overthink it, all right? We know that when you build nucleotides, you can only add things on the 3' end of an OH. This thing was extracted out of some sample somewhere, and it just happens to have phosphates on both sides. That's it, okay? So this can be found in DNA. The above thing has a 3' OH. Is that true? It's actually false for this example, because what's on the 3' end in this picture? The 3' end on this picture specifically is a phosphate. So this does not have a 3' OH on here. There's a phosphate in this picture. That's why you got to interpret the picture instead of just memorizing facts. You also have to interpret what is given to you. Okay? So this would be, yeah, it has a 3' phosphate, not a 3' OH. Other things they could ask about this is, what if you use a 5'A exonuclease on this thing? Okay, so if you don't remember, an exonuclease, what its job is to remove nucleotides one at a time from one end of this DNA strand. This number here tells you what end it will remove nucleotides from, and this letter will tell you how it will do that, okay? So you can have 5'A exonucleases, you can have 5'B exonucleases, you can have 3'A exonucleases, and 3'B exonucleases. I'm just going to do this first one here. And here's how it works. So I'm going to redraw this DNA sequence like so. Right, and here's all your phosphates in there. These phosphates in the middle are usually not drawn for you in a DNA sequence because they're just part of the backbone of a nucleic acid. But it's important, and you may want to draw this really quick to help you answer a question like this. So if I use a 5' A exonuclease, the 5' part tells me that is going to be removed. The front base, the base on the 5' end of this DNA strand will be removed in some way. The A part tells me it will do so by cutting before a phosphate, the left side of a phosphate. So if I cut the left side of this phosphate, I haven't removed anything yet, so it doesn't count. But if I cut before this phosphate, I will have removed this thing. Basically, the fragments we're going to get is A with a phosphate there, and the other fragment there. Okay, so using 5' A exonuclease, you draw it out, and you report what fragments are made. Yes?
So what if it's a 5' B? Would you get P and then A and everything, or would you just get P?
If it's a 5' B exonuclease, its job is still to try to remove the nucleotide. So if it only removes the phosphate, you don't count that. You have to go to the next one. So yeah, for an example, just for an example, let's do 5' B for this example up here. If we use 5' B, it'll still try to remove the A. It will do so by cutting after a phosphate, so you end up with
So it wouldn't
this.
try to remove before the A at all?
No, because its job is to remove the nucleotide, not the phosphate. So it will actually cut it like that. Okay, that would be for 5' B exonuclease. So I won't go over any more because there could be, one of those could be on the exam. But those are two examples for you, yes?
So basically, we would obviously have to know how to draw like the sequence. Okay?
It does help a lot, yes. Some people actually do choose to just memorize, yeah, if it's 5' A, I automatically get that. I always have to draw it out and draw the cut itself. Okay.
I have a
Yes?
We had questions on the practice exams about endonucleases as well, so I don't know.
So, endonucleases, there was a long time ago, I don't know which semester, he tried to kind of throw a curveball at you guys by saying, if I use a endonuclease, what will happen? One, you should know that an endonuclease, what that will do is it will cut somewhere in the middle of a DNA sequence. You won't have to know where in the middle, but it will cut somewhere in the middle of a DNA sequence. The key for that, and it's probably not going to show up this semester, the key for that is the fact that he would have to tell you if it's an A endonuclease or a B endonuclease. The A part will tell you it will cut before a phosphate somewhere. The B will tell you it'll cut after a phosphate somewhere. So you inspect the fragments and you can kind of figure out which endonuclease was used. Yes?
Would he have to tell you if it's 5' or
An endonuclease doesn't work like that. An endonuclease looks for a pattern.
So it would only be like, if you said G,
Mm-hmm.
so it would only be like, since there's only one G in like the middle, it wouldn't be able to do it if there were two G's, right?
Endonuclease is more complicated than that. That's why it doesn't really show up on the exam like this question anymore. But so don't worry about it too much. 1 second. Yes.
For the 5' exonuclease,
Mhm.
wouldn't it cut to the right of the phosphate, so there wouldn't be a phosphate on the...
Oh, yeah, I did forget that. Thank you very much. I added too many phosphates there. That is exactly right. That should be an OH there. Exactly right. See? You guys are already ready for the exam. You identify when I make a mistake. All right. There's other questions. Oh, yes. Yes, if there's no phosphate there, what ends up being there if there's no phosphate is a hydroxyl. That is the default functional group that's left over if there is no phosphate there. Back there, yeah. Yeah, he will never draw the hydroxyl. It takes up too much space. Too much ink, I guess. I don't know. But hydroxyl will not be drawn for you on the exam. So you can always assume that if there's an end of a nucleic acid sequence that doesn't have a phosphate drawn in for you, there's a hydroxyl there. And that again applies to these exonuclease questions because the only important part for an exonuclease question is where the phosphate is. Okay. All right. Let's see. Where were we in the PowerPoint? Exonucleases, restriction enzymes. All right. Restriction enzymes. General information is needed for exam 3. All that stuff about recombinase, cloning, and everything that I prepared you for won't be on exam 3. So, I will get to it again in a second. Don't worry. Basically, first, to introduce you to the concept, restriction enzymes. You should know the general information of a restriction enzyme. A synonym for a restriction enzyme is endonuclease. So what a restriction enzyme does is there's a particular pattern. Let me find a nice example, I guess. Visually, this is to illustrate what a restriction enzyme will do. Here's a restriction enzyme called EcoR1. What it does is it looks all across a particular genome given for this specific pattern drawn on here. You don't have to memorize what the pattern is, just know that a restriction enzyme looks for a pattern in the genome, and every time it finds that pattern, it will perform a cut in DNA. And the end result usually is DNA will open up, or a fragment of DNA will be cut out of the genome target. So that is what a restriction enzyme does, is it looks for a particular pattern and it cuts it. That is what a synonym for restriction enzyme is endonuclease, which is what we mentioned earlier. Other general information that's important is, where is it? Here is the general information of like type 2 versus type 3 and type 1 restriction enzymes. It's kind of given to you right here, so I'm not going to go over it in detail, but be able to, in a multiple choice version, be able to tell the difference between them. Really, the important part is this thing here, is a type 1 restriction enzyme is unable, it does not have a specific pattern it looks for, all the other ones do. Let's see. Also, other differences, these are listed on here for you. I'm not going to go over it in detail, but look at the differences between them, and only the information that is given to you on the PowerPoints will be asked about in regards to restriction enzymes. So don't worry if there's something missing here, you won't be asked about it. Yes?
So can it also cut when the sequence is reversed?
Yes, so that usually happens if it's like on the opposite strand. That happens all the time. Yeah. In fact, restriction enzymes require both strands of DNA to match the pattern that it's looking